Just 10 minutes. All right. So let's imagine that's your skin cell, and you get infected by a nail. Let's say you got a nail, it's cut you up a bit, and a pathogen enters. We'll just call it the microorganism. Could be bacteria, could be anything. Then what happens is you've got yourself your phagocytes. That'll be your first defense. There are three that at least we've got, right? So the first one is your macrophage. That's your antigen presenting cell. You've got your neutrophils. These last approximately 5 days. They're very short-lived, and about 60 to 70% of your white blood cells. Their job is just to engulf pathogens. One job, engulf and digest them. They do not present their antigen on the surface. And you have your mast cells, and this we'll talk about a bit later. So this is your mast cell. Okay, so first things first. Once this pathogen enters the body, it gets engulfed via endocytosis by your macrophage. Once it's in the macrophage, it's in a vesicle we call a phagosome. It's in a phagosome there. Then what happens is it will fuse with the lysosome that contains protease, the enzyme that digests your proteins. So these two, the phagosome and the lysosome, they will fuse together and form a phagolysosome. And that's where the antigen is processed. Antigen is processed, and then it's sent up to be presented on an MHC protein. So then your antigen is then presented there. And remember that antigen, right, is then going to bind with your T cell. We'll just call it directly the T helper cell to activate it. So once your T helper cell binds there, it's going to be active. Okay, and this is where now the domino chain reaction happens. Okay? So the T cell releases these chemical mediators known as cytokines, right? So it releases heaps of these, and what that does is it triggers it to divide into your T killer cell. Now T killer cell or cytotoxic T cells have one job. They are going to kill infected cells. So if there's an infected cell, maybe by a virus or bacteria, yeah, what this T killer cell is going to do is going to ask this cell to commit suicide. Programmed cell death, apoptosis. It's going to die. That's all the job of your cytotoxic T cell. Then you have your memory T cell, and these ones stick around in case the antigen reappears. So that's your memory T cell. Okay, so T killer cell, sometimes they call you cytotoxic T cell. Now that's with the help of cytokines. Those are your chemical mediators in red. Okay. The T cells also activate our B cells. So we've got B memory or memory B cells, and they differentiate also into your plasma cells. Okay. Now your plasma cells have a lot of rough endoplasmic reticulum. That's one of your organelles in the cell. Now this rough ER, its job is to create proteins. Now this plasma cell, what it does is create proteins. It will just create this Y-shaped looking antibodies. Heaps of them. That's why it has a lot of rough ER. So it can make many antibodies. So it creates all these antibodies. These antibodies have a few purposes. One purpose is labeling. So how these antibodies work, they will label some cells, like your mast cells. So they will come there and they will embed themselves in the plasma membrane of mast cells like this. So once your mast cell now has antibodies, they are specific to that antigen, very specific. They fit complementary, like a lock and a key. So they do that labeling. Another way, and this is stuff you could add to your answer, another way is they inhibit mobility by attacking the flagella of bacteria. Bacteria have this flagella that gives it motility. All right. Then it can also act as an antitoxin. Bacteria such as Clostridium tetani causes tetanus. It releases toxins. So some antibodies, they can attach to this bacteria and stop them from producing this toxin. The fourth way is agglutination. Agglutination is clumping. Imagine like sticking the bacteria with glue everywhere, and then they just kind of stick together. They can't perform their function once that happens. Agglutination. Okay. So what you want to focus on, obviously, majorly is the labeling, because that's for your mast cells. Add this other stuff for extra marks. Okay. So from there, this specific antigen, okay, that's complementary to the antibody. Once it attaches there, and these antigens will be present everywhere. This is the bacteria I'm drawing. So that antigen that's been processed, right? It will attach to that mast cell. And once that happens, it sends a signal to degranulate. So you have granules in here, in the mast cell like that. Okay? And they are full of histamines in red. So what is going to happen is, I should have used red color. Anyway, they degranulate, open up, and release histamines. And these histamines are responsible for your inflammatory response. That's your itchiness, swelling, fever, all of these things. And what they do is, imagine that's your epithelial cell. Okay? So these cytokines, they create this inflammatory response here. And this allows for our phagocytes, your macrophage, your neutrophils especially, your mast cells, all of these, they will squeeze through this capillary cell. They will kind of squeeze into there by a process called diapedesis. Okay? This just concentrates all of these white blood cells to the site of infection. Okay? So once they're there, then they can ingest all the antigens, all the bacteria, and kill the infection. So that is the main way. Now, that's your UV question, right? You've mentioned macrophage, you mentioned T cells, you mentioned B cells, and you've gone a bit to depth about how antibodies work. And obviously phagocytosis. Okay? So this whole process, remember at the beginning of this macrophages, presenting the cell and ingesting it, it's called phagocytosis. Okay, now let's imagine this. Let's imagine it wasn't a nail. Okay, so now imagine that this was something that causes an allergy, like a bee. Okay? So that's a bee, and it's coming, and it's gonna, provide an allergen there. Now don't confuse yourself. The allergen, antigen, will be the same thing. So this allergen, again, is going to be eaten by your macrophage. Now the question is going to be, why is there delayed response? This is because initial exposure to this bee that you've never been stung from before, it's going to have to go through this whole process of activation, cytokines, memory, T killer cells, memory cells, right? Go through all of this before it can actually go and label the mast cell, which creates these histamines, right? Which is why this delayed response, because of that first exposure, they got to create these memory cells and all of this. Now, let's say that bee, that same bee, stung you after 5 days. Now you had the allergic reaction, right? You had the allergic reaction. Your antibodies did the job. they eliminated that antigen. Your T suppressor cells, don't forget about those. They stop the infection once all allergens are done. They stop the immune response once all allergens are done. Now, 5 days later, that same bee or different bee comes and stings with that same antigen. Okay? What's going to happen is your memory bee and T cells are going to kick in. They will quickly be able to produce antibodies. Right? That will then be labeled very quickly on your mast cells. They'll embed themselves in the mast cells and there'll be histamines produced. Now, imagine if after that 5th day I said the bee bit you again. Now on that 6th day, the bee bites you again. Right? Now what's going to happen is basically you're going to have a lot more of these allergens there. And so there'll be a lot more antibodies being produced, right? And because of those antibodies, there'll be a lot more mast cells labeled, and there'll be a lot more degranulation. So further and repeated exposure of this antigen or this allergen is going to cause a more rapid and more severe immune response. That's the difference between the two questions. Does that make sense? Did I miss anything? I think that's about it. I think I missed the T suppressor cells. So those are the ones that, yeah, no, they stop the immune response once it's done. Yeah. Good.